All right, in this section, we're going to discuss osmosis. Osmosis relates to the diffusion of water. Osmosis is the net diffusion of water molecules through a semi-permeable membrane. Like all diffusing molecules, water molecules move down their concentration gradient from areas of high concentration to areas of lower concentration without an input of energy. Water concentration is lower wherever solute concentration is higher. The presence of solutes excludes some of the water. Therefore, osmosis can be defined as the diffusion of water toward higher solute concentrations through a semi-permeable membrane. The concentration of solutes on either side of the membrane determines the net direction of osmosis. Osmosis will take place whenever the solute concentrations on either side of the membrane are unequal. Most cells have aquaporins in their cell membranes, which allows the rapid diffusion of water to maintain dynamic equilibrium. Solutes may be excluded from the membrane, or they may also be moving across the membrane, impacting the net direction of osmosis in a dynamic fashion. To begin our study of osmosis, consider solutions containing solutes that cannot cross the semi-permeable membrane. Models of osmosis. We can study osmosis by considering fictitious beakers that are bisected by semi-permeable membranes. So you can see there's two fictitious beakers here separated by this red dotted line that we're imagining to be a semi-permeable membrane. Here, water flows from the compartment with a lower concentration of solutes. In this case, on the left, the water will flow from the left into the compartment with a higher concentration of solutes. You can see the water increasing on that side. The flow of water will continue until the concentrations are equal and a dynamic equilibrium has been achieved. So the concentration of the solute in the water on the left is now the same as the concentration of the solute in the water on the right. At first, the net flow of water is from the left to the right compartment. Once equilibrium is reached, the flow of water is equal in both directions. That's what makes the equilibrium dynamic. The initial rate of net diffusion is high when the concentration gradient is high, and the rate decreases as equilibrium is approached. Example 1, osmosis. Another model used to learn about osmosis is a U-tube, which you can see here, which is also bisected right here by a semi-permeable membrane. So you can see we have this gray is the water, and the red dots are the dissolved salt, the solute. So the question we need to answer is, what will be the net direction of the water flow? And what will the U-tube look like at equilibrium? So before I go to the next slide and review the answer, remember that the osmosis will be the flow of the water toward the higher solute concentration. So the water is going to flow from right to left towards the higher solute concentration. Okay? So on the answer slide, I've drawn sort of what we might expect to happen. So the water will flow from the right to the left because we had more solute particles on the left. And at equilibrium, the water level will be higher on the left side and the concentration of solutes will be equal on both sides. Osmosis in biological systems. Polar water molecules diffuse very slowly through the lipid bilayer and very rapidly through aquaporins. Aquaporin protein crosses the phospholipid bilayer 6 times with alpha helices that create a hydrophilic water-specific channel through the membrane. So this is what the aquaporin channel looks like. It's a channel for the water to be able to go through, because remember, this inside of the bilayer is hydrophobic. So water can pass through, but not very easily. But if we make a channel that has a hydrophilic inside, then the water can pour through. The alpha helices of aquaporins are what create the hydrophilic water channel. Example 2, osmosis. Cells have an average solute concentration of approximately 0.15 molar, which equals a 0.9% sodium chloride solution, which would be 0.9 grams of sodium chloride in 100 milliliters of water. What will happen to these red blood cells if they are put into pure water with no solutes? All right, so if cells that have solute in them are placed into a water solution with no solute at all. So if this is the membrane, on this side we have solute, on the outside we have no solute, right? What direction is the water going to flow? The water flows toward the area with highest solute concentration. So the water is going to flow toward where there is solute. And what that means is water will rush into the cells through aquaporins. The cells will swell and will eventually lyse or burst. Remember, animal cells have no rigid cell walls to protect them from lysis. So if we place these cells into pure water, we will get swelling and lysis of the cells.